The next part of forensic science deals with DNA analysis. First, let us determine the application of DNA analysis in forensic science. DNA profile makes it possible to associate a DNA sample to a specific person with high degree of certainty, thus giving law enforcement and forensic science a new and powerful identification tool that complements fingerprints and other methods of identification. Next, let us have a review regarding the nature of DNA. DNA is a molecule that is found in nearly all cells. Notable exceptions are red blood cells, which have no nucleus. DNA is a special type of molecule known as a polymer, which is a molecule made up of repeating simpler units called monomers. DNA is located in two regions in a cell. First is the nucleus, and second would be the mitochondria. Both can be used in DNA typing. However, mitochondrial DNA is of a different length and shape. Unlike nuclear DNA, mitochondrial DNA is inherited only from mother. It is used for typing under different circumstances than nuclear DNA. Nuclear DNA is found in a geometric shape called a double helix. A helix is a spiral shaped object. DNA looks like two helices that wrap around each other. Consider a ladder made up of two poles held together by a series of rungs. To recall, when an adenine base and a thymine base come into proximity, they form a bond to each other. Likewise, when cytosine and guanine get near each other, they will bond. The order of these pairs of bases is the principle of genetics and inheritance. The order of the base pairs contains a sort of blueprint or genetic code that determines many of the characteristics of a person. Dr. Alec Jeffreys applied the technique of restriction fragment length polymorphism or RFLP to a real criminal case and showed that this technique could associate a person to biologic evidence through DNA typing. But it is not used any longer in forensic applications, having been surpassed by methods with higher powers of discrimination and whose results can be obtained faster using much less biologic material. For a while, many forensic science laboratories use both RFLP and PCR methods in tandem. Today, most laboratories use a typing method known as short tandem repeats or STRs, which combine some of the attributes of both PCR and RFLP. So the first method of DNA testing is restriction fragment length polymorphism. The length polymorphism present in the variable number tandem repeats or VNTRs is used to discriminate a population of people. The use of RFPL typing in forensic science has decreased in the recent years in favor of PCR-based methods, but it is still used in some forensic laboratories that perform paternity testing. Around the same time in the 1980s that RFLP was being developed for forensic use, another major advance was the development of forensic applications of the polymerase chain reaction or PCR. PCR overcame one of the major problems facing forensic biologists, which is that in RFLP methods of DNA typing would require relatively large quantities of DNA. So these are now the steps involved in PCR. First is denaturation. As the double-stranded DNA denatures, the bonds between the base pairs that hold the strands together would break, resulting in single-stranded DNA. So as long as this temperature is maintained, which is 95 degrees Celsius, the strands will remain apart. The next step in the PCR process is annealing, which is the attachment of short strand of synthetic DNA to each of the separated strands. These are called the primers because they will mark the starting points for the addition of new bases to complete the reproduction of each strand. The third step is extension. Under the influence of Taq polymerase, single bases are added to the primer one by one. Each base is complementary to a single nucleotide present on the strand being duplicated. The entire complementary strand is built up and a new piece of double-stranded DNA is produced. This process occurs at each of the complementary single strands created by the denaturation process. So the end result is that two identical pieces of double-stranded DNA are produced. This completes one PCR cycle. The two strands of DNA denature forming four single strands are in turn subjected to annealing and extension, forming four new double strands. The process continues until a sufficient amount of DNA is produced, typically 25 to 40 cycles, which takes about 3 hours. So this would illustrate the PCR amplification process. Each cycle doubles the amount of DNA. Under ideal conditions, 30 cycles would produce over 1 billion copies and take about 3 hours. After the completion of the amplification process, the products must be detected. There are a number of ways to do this. The first DNA region widely subjected to amplification and typing for forensic purposes by PCR is the HLA or human leukocyte antigen DQ-alpha, which is now called DQA1 gene. This gene exhibits sequence polymorphisms. Neither DQ-alpha nor polymarker DNA typing is used in forensic science anymore. These methods have been largely surpassed by STRs. One reason for this is that the alleles in these markers do not vary to a great extent in the human population. As a result, it is not possible to generate a DNA type that is rare enough to associate with just one individual. A more important reason is that this method of DNA typing is not capable of resolving multiple DNA types that are present in mixtures such as vaginal swabs obtained after rape cases. Finally is the short tandem repeats or STRs. These are the advantages of STRs over the other methods of DNA analysis. First, STR markers exhibit high variability in a population, thus giving rise to high degrees of association of evidence with a suspect. The small size of the repeats makes STRs much less sensitive to degradation of DNA. Unlike in PCR, methods are very sensitive to contamination by foreign DNA. For this reason, DNA extractions are always done in a location physically isolated from the place where the subsequent amplifications will be performed. Finally, there are many microsatellites to choose from for forensic purposes. Thousands of them have been identified and many are used for commercial and medical purposes. There are two approaches to gender identification using DNA typing. On the sex determination chromosome, there is a locus called amelogenin. One of the regions of this locus is 6 base pairs longer in males than in females. Females have two X chromosomes and will thus show only one band for amelogenin. Males have one X and one Y chromosome and will thus show two bands, one 6 base pairs longer than the other. The other approach to gender identification utilizes YSTRs. Y chromosome found only in males, also contains STRs. Why STRs are quite useful when typing mixed samples that contain sperm and are thus guaranteed to have a male fraction. But it should be noted, however, that Y chromosome analysis produces only a haplotype and is thus not as informative as common STR analysis. Other than nuclear DNA, we also have the mitochondrial DNA, and these are the applications in forensic science. Number one, all male and female mitochondrial DNA comes from the mother. There is no empty DNA from the father. This means that, except for the mutations mentioned previously, every descendant of a woman should have the same empty DNA. This makes empty DNA very powerful for tracing family lines back through the maternal side. Next, empty DNA often shows a high degree of variation between unrelated people, making it a powerful tool in forensic typing. However, because there are only two hypervariable regions in empty DNA, the population statistics are not nearly as discriminating as with nuclear DNA. And lastly, because of the large number of copies per cell, empty DNA can be useful in typing samples that have low quantities of cellular DNA or in exhibits that are degraded or very old. Many forensic science laboratories that perform genomic DNA analysis do not perform empty DNA analysis. Those that do empty DNA analysis generally use DNA sequencing. They determine the entire base pair sequence in the two hypervariable regions rather than relying on length polymorphism.